Welcome, Life Sciences, to another exciting life science lesson. Today, we're almost completing our journey with microorganisms. The last couple of lessons, we're looking at this whole concept of what a microorganism is. It's something that's really, really small, but we also looked at the huge impact that it had, on our society. So let's just quickly recap. What did we look at? We started off looking at viruses, then we looked at bacteria, then we looked at protists, and the last lesson would have been on fungi. And one of the things that we concentrated on in those lessons was we concentrated on the structure, the characteristic, and all of those we looked at a pathogen, right? And what is that? It's disease-producing bacteria or disease-producing, protists. And we concentrated a lot on the diseases that these microorganisms caused. Today, we're going to look at it in a totally different light. Today, we're going to concentrate on why are microorganisms so important in our environment. Guys, as some of the things that we've touched on, we're going to briefly touch on again, because at the concept of, today we're looking at functions of microorganisms, and they play such an important role, in our environment. Our question for today, says it. It's that we've concentrated in the last while on these diseases, what makes us sick, etc. And today we're going to take a totally opposite stand, and we're going to say, all right, yes, I know that it causes diseases, but guys, the most important thing is we can't live without microorganisms. They are essential to our environment. Okay, if we have a look at the mind map, it's quite a busy one today. Usually I keep things quite simple, but that also tells you how important microorganisms are. The mind map tells us where we're going, what do we need to concentrate on. As I said to you, a lot of the concepts we might have dealt with, but we're taking all of those importances and we're packing them in one important concept. A lot of the terminology, again, words that you might not recognize, you might not understand. Some of them we have done in previous lessons, so I'm hoping that you remember, that you recall, and you can think, oh, I remember that one. All right, or maybe this time, oh, I forgot about that, and let's go back, check on our notes, check on our textbooks, right. Underline it, highlight it. I need to know these important concepts. As I said to you, life science is a different language. You must be able to speak those terms that we're going to discuss. Okay, so now we're going to start looking at why is it so important, why are microorganisms so essential to our lives. Now, the first term that we're going to look at, is this word decomposers. Now, a decomposer, things die. That's one thing you've got to know, that living things, all living things die. And here, I put a picture of an animal, and here I've put a picture of a plant, and everything dies. Everything, all living things ultimately are going to die. Living things are made up of stuff, They're made up of nutrients. And what decomposers do is they take those nutrients and they return them back to the environment. Guys, you don't drive around or walk around and you see dead things lying all over the show. And that's what decomposers do, Everything that dies, they need to return all those nutrients into the soil. And bacteria and fungi, remember, bacteria and fungi, they are probably two of the most important microorganisms that return these nutrients back to the environment. Now, the next concept you might remember, if you were looking at grade 10 syllabus. Now, the grade 10 syllabus, you might have done it or you might have looked at it earlier on, is this whole concept of nutrient recycling. What do we mean by that? There are certain things, like nitrogen's one of them, carbon dioxide is one of them, oxygen was one of them. Don't know if you can remember, Water is one of them. And all of those things are non-living. And we can't just make them. So what we need to do is we need to recycle. What does recycle mean? It's almost like a continuous circle. We use and we give back. We use and we give back. And that's exactly how microorganisms are going to play a huge, big role. I want you to have a look at this diagram. It might be familiar to you. This is going to pop up once or twice. What is really, really important is this nitrogen. Right, so nitrogen is a very important element, but have a look here. What am I looking at over here? This decomposers. Look at this over here. What do decomposers do? They play such an important role in returning nitrogen back into the atmosphere. Now, when it comes to nitrogen, right, nitrogen, guys, is approximately 78% of the Earth's gases. 78%. That's more than oxygen, carbon dioxide, all of that. But plants and animals, we don't have a way in which that we can take in oxygen. So what do? We rely on bacteria, we rely on fungi, but especially bacteria. And bacteria are able, right, to take the nitrogen and to be able, right, to convert it to something that plants can use and animals can use. And over here, remember what I said, it's recycling. So what do? When an animal dies, what does it return to the soil? The nitrogen, right? It's returned. And that nitrogen changes, and so an animal eats it, the animal dies, it's returned to the soil or the atmosphere, the animal eats it, etc. This whole concept of recycling, of using, giving back, using and giving back. We see it here as well when it comes to carbon dioxide. Maybe you looked at that, right, in previous lessons. All living things are going to give off carbon dioxide. And when do we give off carbon dioxide? When things die, right? So dying things, once again, what are we doing? We use oxygen and we give off carbon dioxide. Plants use oxygen, and when things die, what do? They give off carbon dioxide. So this whole concept of using, returning, using, returning. And that's what microorganisms do, all right? They help return these needed nutrients back to where they need to be. Okay, guys, now it comes to oxygen. Oxygen, exactly the same as nitrogen, exactly the same as carbon dioxide. We can't just make it. We have to recycle it, if I can use that. And how do we recycle it? The recycling comes in, have a look at this color. Right, any kind of organism that's got chlorophyll. Because if it has chlorophyll, what can it produce? It can produce oxygen because it can photosynthesize. Now, if you watch some of the previous lessons, you would have noticed this. What is this? This is phytoplankton. That is tiny little microscopic organisms. They're both protists and they are found right in aquatic mediums. Here, right, this is blue-green algae, right? And some of what are algae able to do, and some bacteria, cyanobacteria have also got green. So we've got bacteria, we've got algae, right? And what can they all do? They can photosynthesize. And what does that mean? They take in the carbon dioxide and they release oxygen, because all living things need oxygen. So it's a regeneration. We can't make it, but we can produce it. Plants can produce it in a concept called photosynthesis. Okay, now we're going to pop up this diagram that I showed you just now, is the nitrogen fixers. Now I'm going to show you the diagram before I come here. It's the same diagram we used just now. So here was my decomposers returning the nutrients, all right, to the soil. And who benefits from these beautiful nutrients? The plants. But now this concept of nitrogen fixing, as I said to you, all right, this gas, so important, yet we cannot use it at all. So what do we have? We have bacteria that live in the soil. Some just living in the soil, some on the roots of plants, right? And what do they do is they are able to take nitrogen, right, from the air, and the bacteria converts it to something called nitrates. And what can the plants do? They can absorb those nitrates in the water, in their roots. And those nitrates go up the plant, and the plant can then convert it to nitrogen. All right. So any kind of plant that we call a protein. I don't know if you can remember biochemistry, but if there's nitrogen present, it's a protein. And that's how animals, right, need to eat the protein, the plants that have been able to convert that nitrogen. Right, now guys, also a concept that we've looked at before. What is this rhizobium? Now this is a bit of a picture over here. Now what it is this is the bacteria, right? This is the bacteria that we find. It's a mutualistic relationship. We're going to see a picture just now. But what this bacteria does, here's bacteria that we just said. Okay guys, if we take a closer look, all right, to what these bacteria are. So here you go to see a picture. Here's the roots. So those little round spots, you see I've labeled them rhizobium. That's basically what the bacteria are, right? They're these little conglomerations on the root. And what they do is, they're not like fungi. As I said, they're bacteria. And what they're able to do is here in the soil, as I said to you, they convert the nitrogen to nitrates. And as soon as the nitrates go in, can you see it right by the roots? The roots can take them up. And where do we ultimately need to go? To the leaves, right? And why do we need nitrogen in the leaves? Because nitrogen is a really important, right, element for the making of chlorophyll. So plants need to be able to use that nitrogen to make the chlorophyll as well. Okay, now one of the concepts that we can use, right, is biological control. I don't know if you've ever heard of this concept before, but very often when we looked at here, let's go to the malaria one. When we looked at malaria, we said the one way of getting rid of that mosquito was to spray with DDT. Okay, now when we spray, we use chemicals, and chemicals can have a very harmful effect in the environment. So one of the ways that we can get rid of harmful things without harming the rest of the environment is called this concept called biological control. Right, so what it is over here, I want you to have a look. Do you see those round brown things on the branch? You see those big round brown things, okay? That is actually a fungus. Now, it looks really gross and it's killing the plant. And we say, yes, right, yay, why are we excited for killing the plant? Because this tree over here is an alien, not as in an alien from another planet, right? What do we mean by an alien plant? It's a plant that lives in South Africa. It's one of an acacia trees, but it's not indigenous from here, right? And it actually causes a lot of harm. It takes in a lot of water, and our country is not a country that's full of water, right? We actually are a dry country. So this plant actually kills the things around it, and it's not good for our environment. So what we want to do is, we don't always want to chop it down because there could be a lot of things in the trees. So what we do is you infect it with a fungus, and that fungus feeds off of that tree, because that's what fungus does, because it doesn't have its own chlorophyll, and it feeds off the tree and it ultimately kills it. So it's fine, all right? We don't want that tree anymore, and we give the fungus an opportunity, right, to feed. That is biological control. No chemicals used at all. Okay, now we're going to look at just a few symbiotic relationships. And if you looked at symbiosis, you know it's when animals or organisms or plants have a relationship, and we're going to pay particular one to mutualism, where both of the organisms benefit. Okay, guys, here in our tummies, not so much our stomachs, in our colons, in our large intestines, we have got lots and lots and lots of bacteria, right, growing in our colons, and that's really cool, all right? Because what happens is that they actually make some vitamins for us. So they make vitamin K especially, so that's how they're going to benefit. But what do they do for us? What's in my colon? Food, right? And what do the bacteria need? they would like food. So I give it bacteria, all right? I give the bacteria food, yay for bacteria. And what does it do for me? It gives me a very helpful vitamin that I need to help with blood clotting. Both parties benefit. This one we looked at, so I'm not going to spend too much time. All right, guys, here's the bacteria on the roots of plants. Right, what does it give the plant? It gives the plant nitrates, nitrogen, nutrients. And what does it the plant give it in return? Food. Guys, plants are food. Right? That's the relationship that they're going to have. Right? Bacteria helps with the nitrogen, and plants say to the bacteria, here guys, have some glucose. Because all cells need to cellular respire, and we need glucose and oxygen for that. Here's a nice concept as well. These are fungi. Again, it's the roots. Have a look at the white over here. So there's lots and lots and lots of white, and you guys think, oh, that's the roots. It's actually not the roots. It's the roots come like this, and all these other ones I'm going to do just show you a different color. Right, those are all fungal threads. Right, and they increase the absorptive area. So instead of having just a few roots, you have all these fungal threads. And what can fungus do? It can absorb water. So what does it do for the plant? Large amounts of, water and nutrients. And what are plants? Plants are food, because the fungus can't make its own food. Right, so they help each other. Right, mutualism, both benefit. Okay, guys, that's enough for now. We're going to take a quick break. We'll be right back. Welcome back, Life Sciences. I hope you took a nice little break. As I always say, get those muscles going, clear that head and all the oxygen going through it. What are we concentrating on today? We're looking at all the good things that microorganisms, what they do in our environment. As I said to you, we concentrated a lot on the diseases, and this time we're looking at what functions do microorganisms have that we cannot live without. Okay, so let's carry on with that. There are a few terms up on the board here again that you might need to be more familiar with. Some of them, might be, as I said, might be strange to you, but as we go through the lesson, I will explain the concepts and they will be a lot clearer, but you must remember them, okay, and you need to learn them. Okay, the first word is biotechnology. And this is a concept you're going to look at much later when you look at, if you do more of the grade 12 syllabus. And basically what biotechnology is how we're able to use living organisms for our benefit. And a lot of the time it's a concept of, I won't say genetic modification, where we change the genes of certain things to be able to help us, right? Or use things in order for our benefit. Okay, what's the first one we're going to look at? Now, the first one is this concept of antibiotics. Now, I know a lot of you, I'm sure, have, let me change the color back again, have had antibiotics, you've been sick, and antibiotics were a huge, big discovery. Now, it was discovered by Alexander Fleming, right? He did a little bit of an experiment. He was actually quite a dirty man, by the way. And what he did was he grew a fungus called penicillin. And he noticed that wherever the fungus was, there was very little bacteria. And this is where the whole concept of an antibiotic is. An antibiotic is a medicine, all right, and initially it was a fungus that they could harvest, that they could treat bacterial infections for. So when it comes to antibiotics, guys, this is very important, bacterial infections. Not fungus, we used a different medicine for fungus, and definitely not viruses. Antibiotics are drugs, all right, that we use specifically to treat a bacterial infection. And at the time when it was discovered, right, there was the world wars were on. And lots of men were dying on the battlefield, not from being killed in war, but from being injured and dying from bacteria, right, bacterial infections. And this penicillin gave a whole new concept to healing, right? So antibiotics help, right, people to heal. But the problem with it today is that we have too much of it, right? Lots of times people are given antibiotics and they don't finish the course. And what happens is they don't kill all the bacteria, and the bacteria are able to mutate and become back, right, resistant. Remember when we looked at TB, right, there are certain strains of TB that are resistant to the antibiotics, right, and the treatment that people need. So when it comes to antibiotics, sometimes we just need to take the foot off the pedal a bit and not always use it for any single time we just feel a little bit sick. But as I said, a huge, big, all right, I won't say invention, finding, helping us be able to cure little things that often killed us. Okay, now the concept of vaccinations. And those of you who are living through the COVID times, it's a huge, big concept that we're looking at. And a lot of you, all right, when you were small, you were immunized. Now, what it is the following. These two concepts are the following. When we immunize you, usually what we do is we take, I'm going to use a virus, for example, we take a little bit of the virus, and we take away the danger part of it, if I could use it. We make it a weak formed, and what they do is they inject that virus into your body. Now, your body has a natural immunity system, and what does that do is a whole reaction takes place. That ultimately what your body does is that it remembers, okay, that virus that came into your body, and it produces something called an antibody. And that antibody stays in your system. So if a virus attacks you, that same one, your body has got a memory. It says, oh, I know this virus. I think I can, all right, I think I can attack it. And those antibodies then, all right, attack the virus, and basically doesn't mean you don't get sick, but it usually means that you're not going to get as sick. So when we're talking about all these vaccines, when it comes to COVID, what it is we're putting something inside us that stimulates our immune system to make a response. And that response is an antibody or something that is going to fight that virus. It's not a cure, all right? And it's not a prevention. It's not going to stop us from getting sick. But what it could do is if we do get sick, maybe it's just mild symptoms. We're not going to be so sick that we might die. And that's the whole concept of vaccinations. And as I said, for those of us going through all the COVID things, vaccinations are huge. When are we going to get them? What are they about? Do I trust it? There's a lot of things to consider when we look at this. And as I said, what is biotechnology? Us making things to help us. So this is the whole concept. As I said to you, we put some kind of the virus into a vaccine. That's what a vaccination is. A vaccination is the actual injection. And it goes into our body and it produces this antibody. Guys, the next concept, all right, called biotechnology, is genetic engineering. What does genetic engineering mean? It means we change the DNA. Now, for those people who are diabetics, for some reason, there's lots of reasons, they don't produce the hormone insulin. All right. And what used to happen quite a few years ago, people who were diabetics, they had to use pig insulin. All right. And as you can imagine, certain, all right, groups, races, religion, pig products are a no-go. Plus also it's a pig and it's a human. So they might not be compatible. So somebody taking the pig insulin might have an adverse reaction. So have a look what happens over here. This is called genetic engineering. So we take a human cell over here and we cut out the gene to make insulin. So what do we use? We take a bacteria, all right? And in the bacteria cell, they've got a little round piece of DNA, and we cut it. So it's easy, it's cut and paste. We cut it out, and what do? We stick in this gene over here. So you see that red? I put it into the bacteria. All of a sudden, the bacteria has got the gene to say, oi, I need to make human insulin. And it does. And bacteria grow so quickly. And what message have they get? Make human insulin. So what are scientists, doctors, pharmacists, all those in the medical biotechnology, they take that human insulin, and diabetics can inject human insulin. Right, so they take it out, put it into a bacteria, which is a microorganism, and there we're able to have proper human insulin for people, and they won't be able to reject it. Okay, guys, I think some of you might be familiar with this concept, right? Making beer, making alcohol, right? Traditional beer. So obviously in a lot of the African cultures, making their own beer, right, is part of the culture. Making your own alcohol, right? When we went through lockdown, I think lots and lots of people, when we weren't allowed to have alcohol, all of a sudden started to make their own alcohol. So why is it on the list? Guys, check at the top there. Yeast. Yeast is a fungus, all right? And when we make any kind of alcohol, if we're making our traditional alcohol, or if we're having a good old Castle Light or a Castle Black Label, if we're having a glass of wine, all right, or if we want to do all this craft beer that we want to do, all of it involves fermentation. And fermentation is a process we're going to look at later, all right, in the series. But we use yeast, we use the sugar, and we put it together, and a natural process that yeast fermentation occurs, and what is the result? Alcohol, right? Alcohol. So a lot of people are reliant on microorganism yeast to have your alcohol, whichever way, your homemade one, be careful, right? Or the ice cold one that you have. Bread. Oh, I love bread. Right, that's why it goes all the way to the hips. Bread. How do we make bread? Lots of recipes for today that says make bread without yeast. But how have we made bread? We use yeast. All right, the microorganism, it's a fungus, and we use it to bake breads. Again, that fermentation process. And a lot of our bakeries, bread. All right, we love bread and jam. Yummy. Okay, this is a food group for me. For those of you, I love cheese. Cheese is the most amazing thing. I cannot live without cheese. And as you guys remember, a cow doesn't just go tonk and out pops the cheese. Right? What do we use? How do we get something so solid? Quite simple. We take the milk from the cow and we put bacteria in it. And that bacteria makes it all lumpy. We call it curd. And we just basically take all the moisture out, and there we've got delicious, all right, lovely cheese. Now have a look at you. Those of you, right, who like blue cheese. Everybody goes, oh, gross, don't like it. So what do? We take the cheese and we put fungus in it. Right, and the fungus gives it the taste and the smell. Some of you don't like it, but it's fine. Yogurt. Right. Yogurt is packed full of bacteria, really healthy for us, gives us that good bacteria that we need, right, in our colon that helps us produce all that vitamin K. So that's how we constantly are using all of these microorganisms. Guys, have a look here. Right, we call it mass or marcy. Some people say, oh, it's cultured milk. It's the same thing. And you can use it in your baking. I know I make a bread. Can you believe it? Yes, love bread. And what do I add? Cultured milk. I don't add yeast, and I have cultured milk. And what is cultured milk and what is a marzi? All it is milk, right, which we add bacteria again, right, and we ferment it. We change it and it becomes quite lumpy, but it gives the food, or you like to drink it, a nice taste. Okay, guys, that's all that we're going to have for now. We're going to have a quick break and we'll be right back. Welcome back, guys. I hope you had a good break. You always need to just get up, get those, I say to you, get those muscles going, get the oxygen flowing. We can't just sit and just take in stuff. We've got to allow our brain, right, to take in all the oxygen. Remember what we focused on today? We're looking at why are microorganisms so important in the environment. And we had a whole look at how we use it, the foods that we eat, how they're important in the environment. Now we're going to have a look at some of the questions that we can ask using these different concepts. Okay, you always like to start off with the multiple choice. The answers are there, so it takes a little bit of pressure off. And our question is, the use of antibiotics, remember that's what we looked at today, it's one of our biotechnology, is an effective treatment for. And now it gives us a whole variety, right, of answers. The first one, viral infection. And we say no, right, because remember, I'm going to say it over here, antibiotics, we need to jump to that concept that antibiotics are used for bacteria. All right, question number B, all right, is bacterial and protists. So although the bacterial is right, it's only bacteria. The protists, that's going to be your malaria, and that's not, we're not going to use antibiotics there, we're going to use different kinds of medication. So B is incorrect. C, bacterial. Yes, we know it's bacterial. However, we've already ruled out that viral is incorrect. So C has to be incorrect. Question or answer number D, bacterial infections only. Yes, right? That is our answer. Our answer is going to be number D because we're going to use antibiotics for bacterial infections. Okay, guys, our next multiple choice question is about vaccines. Vaccines are effective in protecting the body because, again, why are vaccines so important? What do they do? Right, number A, they trigger the immune system to produce antibodies. Yes, they do. And to protect the body from disease-causing agents. Yes, they do. So what we're going to do is we're going to leave number A. Looks like it's probably the correct answer, but I need to carry on with the rest just in case there is a more correct answer. Number B, they detect disease-causing agents as soon as it enters the body. No, guys, they can't. They've got to take a while. They've got to recognize it. So it's not going to be number B. Number C, they help the body excrete. No, excrete means that's urine or sweat. You can't excrete a disease-causing agent. Okay, so the terminology there is also totally incorrect. The next, number D, allows the disease-causing agents to multiply. Oh, no, 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 no. That's the last thing we want them to do, is to make more of themselves. So number D, all right, is definitely correct, which leads us back to our very first answer, A, which we thought was correct. We just double-checked, and our answer then would be A. Okay, our next question also is a multiple choice question. And this time we have a diagram to go with. The question says the diagram below illustrates activities taking place in the human body. All right, guys, so if we have a look at the questions that we asked. Right, vaccinations, antibodies. So that's the word we are concentrating on. And antibodies, all right, is C. So that's the letter that we want to most probably be the answer. Let's look at our options though. Number A, structure A only. Now structure A is the white blood cells, and we know no, we're looking for antibodies. Structure B only. B is the antigen. No, we're not looking for that, all right? The antigen is already what is on the cell, and that's what the virus is looking for. Do I connect? So definitely not. Number C, now we're looking here for the first time. Structures A and C. Now I said A only was no, but now I've got C over here, which means antibodies. Yes. Now why would A be correct? Because if I have antibodies, I also want to increase my white blood cells to come and destroy. All right, so yes, this is looking like the right answer, but I'm going to go to number D because it structures A, B, and C. But guys, it can't make number B. It doesn't make the antigen. The antigen is already, all right, on the cell. So we are going to look, D then becomes incorrect. So our most correct answer is going to be number C. Okay, the next question, lots of questions on this section in microorganisms involve some kind of reading piece. The question here is on penicillin. All right, so we see penicillin. Why is it a microorganism? We know it's a fungus, and we know, all right, it was an antibiotic. So now let's read what it says. Penicillin was discovered by a scientist called Alexander Fleming in 1928 while he was working with bacteria growing on a nutrient medium, agar. That's what you use for bacteria to grow. He noticed that on one of the plates there was a fungal growth. Around this fungal growth was a clear area where no bacteria were growing. And I'll show you the diagram that attaches there. The bacteria had been affected by something. Remember 1928, they weren't quite sure what this was. That had been released by the fungus. The fungus was identified as penicillium. Okay? Tests showed that penicillium was not toxic to humans. Some people are allergic, but most people it's okay and could be used to treat bacterial infections. Okay, here is his famous plate. There is the fungus at the top, and obviously if you have a looking at the clear area, that clear area over there, no bacteria, fungus was able to stop bacterial growth. Okay, so let's look at the questions. Give the generalized term used for medicines such as penicillin. What do we call those today? Those are called antibiotics. Right, remember we don't use penicillin. There's a variety of ways that pharmacies can now manufacture these antibiotics. Name 2 diseases that can be treated by using penicillin. So guys, we are looking, all right, for any 2 bacterial diseases. Now let's go back to the bacterial diseases that we looked at when we looked at our diseases in the previous one. The first one, most important that we looked at TB. Now the other one that we looked at was cholera. All right, I don't know if you guys can remember when we drank water that was filled with bacteria that gave us a runny tummy, etc. Right, a few other things. You can think of any bacterial disease. Some of you, right, might think of pneumonia. That's what you get, all right, in your lungs, a bacteria that grows in your lungs. So any 2 diseases, but they must be bacterial. You cannot tell me HIV. You cannot tell me chicken pox. You cannot tell me all of those because those were all viral. So we're looking at any kind of bacterial disease there, and only 2. Okay, in some areas of South Africa today, TB medications, guess what, guys? The very next question actually gave you the answer. So if you were wide awake and you forgot about something, you're like, oh, okay, it gave me one. Are not curing some patients of this crippling disease. Why do you think this is the case? We spoke about it earlier. We spoke about this concept called antibiotic resistance. That is when bacteria, certain bacteria, do not respond. All right, they do not respond to antibiotics. So we can't stop them, and they keep on multiplying, and they can make us sick. So this is, what do you think? Why do you think this is the case? And it's called antibiotic resistance. Now the next question asks us, remember we often asked you about the management of a disease. What recommendations would you give a TB patient about taking their medication? Okay, guys, when it comes to TB, remember, all right, how do we stop resistance? Very simply, complete the course. Right, you cannot stop midway. You've got to make sure that you finish. And when it comes to TB, finish all the antibiotics, right? Anytime, not only TB, any kind of antibiotics, you must always finish the course, because what you do then is you kill all of the bacteria, and you don't allow some to stay behind and the ability of that mutation to take place. So what would you recommend? All right, complete the course, you finish it. You don't stop when you feel better. That's the biggest problem. They stop when they're feeling better, then they stop the medication, and the bacteria is not, all right, has not been taken care of. This is a bit of a tricky question. You've got to pay attention here, and you've got to look at your diagrams very carefully. Okay, let me read first, and then we'll go and we'll look at the diagrams. Each of the dishes in the experiment was streaked with 5 different types of bacteria, A, B, C, D, and E. Now I'm going to go to the diagram now, because otherwise we're going to get confused. I want you to have a look at this. All right, so the A, the B, the C, the D, and the E, those 5 arrows are the bacteria. All right, so guys, that is really important. Then what you have, let's go back. One strip containing 4 different antibiotics, P, Q, R, and S. So that strip that you see across here, that strip in each of those, all right, that I'm going to put here an A, so your big capital A, so that you know that that strip is A for antibiotic. So each of them have got 4 strips of bacteria, and the dark black strip, that is going to be the antibiotics. Okay, and was then placed across the center. We saw that. The diagram shows the results after incubation for 3 days at 37 degrees. What do we know 37 degrees? All right, body temperature, and that is the ideal growth for bacteria. Okay, so here we've got, all right, here we've got the diagram, a little bit confusing, but let's read the questions. Which antibiotic was the least effective? Now, what are you looking for? You are looking for the antibiotic that grows over, all right, grows over the strip. So which antibiotic was least effective? Here we had B and D, C, D, and E, B and C, and B and E. Which antibiotic was the least effective? So if you have a look here, which this one had 1, 2, all right, this one 1, 2, this one 1, 2, but this one had 3 bacterial growths. So which one was it? Antibiotic number or letter Q. Right, it let 3 go. Which type of bacterium was sensitive to all 4 antibiotics? In other words, which one did which A, B, C, D, or E did antibiotics kill everywhere? I want you to have a look. Look at A there. Look at A there. Look at A there. So the bacteria that was easy to clean, and we used a small a because that was the letter that we used. Guys, which type of bacterium was the most resistant to all of the antibiotics? In other words, which one did grow the most? So if we have a look, it says it was most resistant. So here, if we have a look, what did we have? B and D, C, D and E, B and C, and B and E. Have a look. B appears 3 times, so that means the antibiotics didn't, all right, cure it at all. Okay, guys, I'm afraid that's all we have time for today. I hope you have a greater understanding of why microorganisms are so important in our environment. Until next time, cheerio, bye.